Here in problem 1, we're doing a simple monohybrid cross where we start with an individual that's homozygous dominant, big T, for tall, and heterozygous, big T, little t, also tall but the heterozygous genotype. And the idea is to cross these two individuals, setting up a Punnett square that follows the law of segregation, where each gamete has only one allele. We fill in our Punnett square with the appropriate alleles going down and across, and we end up with our F1 progeny. The question is asking what's the phenotype of the progeny. This individual, homozygous dominant, is going to be tall, and these individuals that are heterozygous are also going to be tall because they have one dominant allele. So the phenotypic ratio is 4:0, tall to short. In problem 2, we're now looking at a dihybrid cross where we're looking at two traits. And we're going to be setting up a Punnett square, again using the law of segregation, where each gamete, we will segregate the big T and the little t alleles, for example, with this first individual. But now we have a second set of alleles, and we have to follow the law of independent assortment as well as segregation. For the law of independent assortment, then, it states that all possible combinations or gametes can be formed. And so here you can have all possible combinations of big T, big R, little r. For the second individual, you again follow the law of segregation, so you only have one allele per gamete, and the law of independent assortment. But in this case, there's only one possible gamete that can form, and that would be little t, little r. There's no need to fill out the whole 16 square Punnett square if there's only one possible gamete. You can save yourself some time. Again, filling in the Punnett square, these are the possible genotypes of the progeny. But the question is asking to determine the F1 phenotypic ratios. This individual is going to be tall, as is this individual, because they each contain one dominant allele. These two individuals will be short because they're homozygous recessive. Then if we look at, for the case of the round versus wrinkled, round is dominant, big R, so this individual is going to be round, as is this individual. These two individuals then will be wrinkled because they have two recessive alleles. So our ratio is 1 tall and round to 1 short and round to 1 tall and wrinkled to 1 short and wrinkled, or a 1 to 1 to 1 to 1 phenotypic ratio. Problem 3 is also a monohybrid cross, but it's asking you to work backwards a little bit. Given the phenotypic ratios of the progeny, 1 tall to 1 short, we need to determine what this particular parent was, what the underlying allele is. We know this individual is tall because it has at least one dominant allele, but it could be either homozygous dominant or a heterozygous. We know that the other parent is short, and so when we set up our Punnett square, we have big T blank, little t, little t. Now we know we have a ratio of 1 tall, both of these individuals are tall, to 1 short. So we have 2 talls here, both of these individuals have to be short. In order to be short, they must have a little t, 2 little t's, and so the little t must come from the parent. Problem 4 is simply the same as problem 3, except for we're given different phenotypic ratio for the progeny, but we would set it up the exact same way. In this case, however, they're all tall for the progeny, so these two are tall, but these two must also be tall. The only way they can be tall is if they inherit a dominant allele from their parent. So the missing underlined allele is a capital T. Problem 5, we're again looking at a dihybrid cross, much as we did with problem 2, and we're looking at, again, the F1 phenotypic ratios. So we start with the first individual, big T, big R, and that's the only possible gametes that can form from this individual. The second individual has, however, all possible combinations of gametes. So again, filling out our Punnett square, and you can simplify it without making the whole 16 square Punnett square by only including the one possible gamete the first individual can form. And we're looking at the F1 phenotypic ratio. This individual is tall, as are all of these, because they all have at least one dominant allele. And similarly, all the progeny are also round because they have at least one dominant allele for that trait. So we have 4 with no other possible combinations, or 100%, you can also say 100% are tall and round. Here in problem 6, we're also looking at the dihybrid cross, but in this case we're actually wanting to determine the F2 phenotypic ratios. However, we're starting with the parental generation, so we first need to determine the F1 ratios. The only possible gametes these parents can form look like this, and so the only progeny that can form from this cross is a double heterozygous individual. We then can cross 2 double heterozygous individuals to determine the F2 ratios. So here, when we set up our Punnett square, we have all possible gametes that can form for both parents. And so this, for the first time, we're doing the full 16 square Punnett square. Now that the Punnett square is complete, we can then determine the F2 phenotypic ratios. The possibilities that we have are tall and green, short and green, tall and yellow, short and yellow. So looking first at tall green, they have to have at least one dominant allele for both traits. We end up with 9 individuals that are tall and green. Short and green, then, we have 3. Tall and yellow, 1, 2, 3. And short and green, we have 1. You'll notice we end up with a 9:3 to 3:1 ratio. Anytime you crossed 2 double heterozygous individuals by each other, you end up with that typical 9:3 to 3:1 ratio. Problem number 7 is looking at a sex-linked trait. So in addition to the alleles for each individual, we also have the sex chromosome designation. The first individual here is the male, and since the trait is only present on the X chromosome, there's no allele designation for the Y chromosome. In this case, the X chromosome has a recessive allele. The female then, XX, is a homozygous dominant with a dominant trait. In this case, for example, we might be looking at eye color, where red is dominant over white. So big R stands for red. We'd set up our Punnett square the exact same way as we would the other examples, except for we include the chromosome designation. And it's typically shown as a superscript. So our resulting F1 phenotypic ratios and sex ratios, and you typically include them together, would be 2 red-eyed females to 2 red-eyed males. Problem 8 is also a sex-linked trait, and we would set it up the same way. The male and female, except for this time the male has the dominant allele and the female has the recessive alleles. The question, though, is asking for the F2 phenotypic ratios, and this is actually the first filial generation. So after we determine the F1, what we'll have to do is cross an F1 female, and you'll notice that both of the F1 females have the exact same genotype, so it doesn't matter, and the F1 male. And again, the F1 males have the same genotype, so we'll have one F1 female by one F1 male. So now in order to determine the F2 ratios, we again cross our F1 progeny that we determined before. And we're now looking at the F2 phenotypic and sex ratios. So we have 1 red-eyed female, 1 red-eyed male, 1 white-eyed female, and 1 white-eyed male. So our ratios are 1 to 1 to 1 to 1. We're going to go out of order here and do problem 10 before we do problem 9 because 10 is a little bit more simple and will help you understand problem 9. Here what we're looking at is the phenotypic ratio of the progeny, and we have to work backwards to determine the genotype of the parents. So starting with what we know, 3 to 1 ratio, should clue you into 4 square Punnett square monohybrid cross, where we know 3 of the individual progeny have at least one big G because they have the dominant trait, and 1 progeny has the 2 recessive alleles. Working backwards then, we can fill in that these 2 parents have to have at least 1 little g to provide the alleles for this recessive individual. And then that means this has to come from this parent, and this has to come from this parent, which means this has to be a big G to provide for that, and this one then has to provide a big G for this. And so we end up with 3 green to 1 yellow, with the parents of the progeny both being heterozygous. And again, if you see a Mendelian ratio of 3 to 1, it should clue you into the idea that it is 2 heterozygous individuals being crossed in this monohybrid cross. As I mentioned for problem 10, it's really a setup to help you solve problem 9. So we're going to do, again, this is in the reverse order where we already did problem 10. Again, we're looking at the phenotypic ratio of the progeny and trying to determine the genotype of the parents. Here, the clue is a 1 to 1 to 1 ratio. This should clue you into the idea it's a 1 by 4 Punnett square. A second alternative is a 2 by 2 Punnett square, and you could solve it that way as well. So we know our progeny are tall and green, tall and yellow, short and green, or short and yellow. And so, working backwards, we know that these gametes have to have little t, little g to make this individual. And so the other little t here has to come like that, and we would get a big G from that parent. And so looking at the genotype of the parents, in order to form this gamete, the only possibility is little t by little g. And to form these 4 gametes, we have to have a double homozygous individual. So those are the two genotypes of our parents. Again, you could have solved it in a 2x2 fashion, which would result in different results. Either way is fine. This is just one example. Problem 11 is looking at some phenotypic ratios of progeny that don't exactly follow Mendelian ratios. So here we're looking at progeny that result in red flowers, pink flowers, and white flowers. With a Mendelian segregation ratio, you might expect a 3-to-1 ratio of red to white, dominant to recessive, but instead we get this intermediate phenotype with pink, which is intermediate between red and white. This should clue you in to the idea that we're talking now about incomplete dominance. So here in this cross, we'll be looking at two heterozygous individuals being crossed to one another, and we'd expect 3 red flowers to 1 white flower, but instead we see 1 red, 1 white, and 2 pink. And so incomplete dominance results when red is incompletely dominant or over white, and you end up with an intermediate phenotype. Problem 12 is looking at the types of gametes that can form from a single individual, so it's not actually carrying out a cross with the Punnett square. Instead, we're just following the laws of segregation and independent assortment to look at the gametes that can form with an individual of 3 traits. Again, following the law of independent assortment, each gamete will have 1 T, 1 G, and 1 R, and again, because of independent assortment, they'll have all possible combinations. So we have 4 possible gametes that can form.